Hello and welcome everyone in another episode of Researcher Celebrity. Today we have Dr. Debojyoti Day with us. A brief introduction of Dr. Day. He has done his bachelor's in technology in biotechnology from West Bengal University of Technology. For master's, he did his specialization in biosciences and bioengineering from IIT Kanpur. For PhD, he moved to South Korea and attended Samsung Biomedical Research Institute, where he did his brief postdoc and then received a tenure track faculty position. But with the heart core belonging to India, he moved back to India and is currently an assistant professor in Department of Biotechnology at National Institute of Technology, Durgapur. With this brief introduction, I would like to say thank you so much, Dr. Day, for taking time out this conversation.
My pleasure. My pleasure. Yeah.
We always start our conversations by one simple question, that how and when you decided that you want to become a researcher.
Oh, yeah. It goes back right from my childhood, my schools. Actually, uh, when I was in high school at the ninth and standard, I was really fascinated. And of course, the thank goes to my physics as well as the biology teacher, who were very kind and were very successful to develop the inquisitiveness. They used to tell us stories of great discoveries, obviously in a way that we understand, and also about the great scientific personalities. At that time, it used to appear some awesome magical world that is going there, that is happening outside the realm of the school education. And that gave me the feeling how fast I can finish this and jump into the research world. And that would be the only my thing. And that's what I am meant for. So that's how it started. And special thanks to my physics and biology teacher at my schools, who were the great catalyst for me to understand and realize this is the only thing I can do and I want to. And that started the passion within. Yeah, it
Nice.
may sound very cliched, but that's what it worked for me. Like
No,
that's
I
how it
think most
happened.
of the conversations, this role
Yeah.
of school teachers is so integral and crucial that they, as
Yes.
you mentioned, like they ignite your brains to think that
Correct.
inquisitiveness, that
Correct.
having unexplored, when the students are just mesmerized by the stories, the discoveries which these
Yes. Yes.
teachers are bringing to the class, which is probably not part of the curriculum, but just to
Yeah.
give
Yeah.
you an idea that what is happening outside world in the real time, especially with the
Correct.
technology what we have these days. So
Correct. Correct.
you already made up your mind in this high school with special emphasis for your physics and biology teacher that
Correct.
sometime you really want to do a PhD. Was that the first path you already had, or there were different thoughts?
Not really. So the first thing was I want to study science in greater depths. Okay. So that was my primary idea. And eventually I realized with some members of my family who were like, for example, my maternal uncle at that time, he was doing PhD when I was in high school. And he used to visit our home and we had great time together. He used to tell his journey about how, like, after schooling and all, how, and of course college, the basic education in the college, how he chose to do PhD and how this path paves the way for him to get a direct access to the enormous scientific world and to do research his uh, of his likes, like whatever he wants to do, whatever he wants to pursue. I say, okay, bingo, that's the way, that's how I should follow. So he was the second catalyst who gave me the idea that PhD is the pathway, and that will give me, because that will obviously entitle me to have that capabilities and to plunge into the world of science and technology. So that is second catalyst that has made this happen. So my maternal uncle was, yeah.
So let's talk about having this background already, that at some
Mm.
point of time you want to explore science, do fundamental or foundational research. What were the key factors when you were choosing or picking up the institutes or universities to do your PhD?
Acha, PhD. Okay, so that is another very interesting story. So during my masters, my project, that the master dissertation, was mostly in structural biology. So I was looking at the structure and the function and the correlation and all of those things. So eventually, I was looking for a position of PhD for a lab who works on structural biology. So that is what, like, I thought would be easier to get, because I had a little bit background, whatever it is, on structural biology. So that is how I started searching. And eventually I got this uh, advertisement, probably in Nature, because at that time Nature used to, and also now also, some Nature portfolio gives the open positions, about the open positions. And I found that in Samsung Biomedical Research Institute School of Medicine, they are looking for graduate students. And when I went there, I searched, there was a very famous lab. My my PhD professor, who was like, his lab was structural biology, and he was famous for a very interesting thing. So as you know, that DNA structure, the A, B, and Z form, obviously the B DNA and the A DNA, all those things were the profound work by Francis Crick and Watson, made it, like, the crystal structure and all those things came to the world. Along with that, there was another interesting part of DNA structure discovery, which was not quite popular, but it is popular among the structural biologists. And that is the structure of ZDNA. Okay. So, ZDNA, the main work on ZDNA was done by Alexander Rich, the famous scientist. So, he was no longer with us. He died at the very old age of 95, 98, I guess. So he was one of the pioneers who was working with the zDNA structure. And interestingly, he was the student of Linus Pauling. Okay, so that is the lineage. And they were all peers, so Watson, Crick, and Alexander Rich. And my professor eventually was a postdoc under this Alexander Rich. And while he was completing his postdoc, he started a very interesting project of understanding how ZDNA, the zDNA, exists within the physiological state. And that is interesting, why I should say, because ZDNA is non-physiological. It happens only outside the cells. But he had an idea that probably it has more than that. It is not only a non-physiological, artificially created structure, but probably it can also be present within the cell. And because there were zDNA binding protein that was already discovered, those proteins which can specifically bind to zDNA with a more affinity than bDNA or other kinds of DNA. So he came back to South Korea. He started working on finding what are the physiological states where this zDNA can exist. And eventually he had an idea that if zDNA exists within the cell, within the nucleus, at some part of the DNA strand is forming zDNA, it also exists as a junction between zDNA and bDNA, because the entire genome cannot be in the zDNA form. So he started working on the concept of B and zDNA junction. And eventually he solved the structure of that junction. very, very crucial, and it became a nature cover page. That was a huge discovery. And also, Alexander Fleming was a part of that paper, that manuscript. And that's how that group, my professor's group, came into limelight, because it was the first time the and the B DNA junction was discovered. So how it may coexist rather, within the cell, and what are the physiological and biological implications. So that's how it became famous. And as soon as I see, oh, this group is actually looking for students, so why shouldn't I be applying to them? So I started applying, and he was very kind. He replied me instantly, and then he set up a call, and we got an interview, and then eventually got into that lab. So that is how I ended up in this lab. But the journey from there was again very different. So yeah,
Absolutely. We
yeah,
will talk about the journey of PhD for sure, but let's
yeah.
understand, and for the audience for awareness purposes, if I can ask you the role of IIT Kanpur has played,
Yes,
the training you received at IIT Kanpur, and to land you in the Samsung Biomedical Research Institute.
yeah. So in IIT Kanpur, the best part was that first of all, uh, we had 1.5 years dissertation. Okay, the first semester was coursework. The next 1.5 years was dissertation. And within the dissertation was so emphasized that we were encouraged to formulate, think, and design problems. May it be wrong, it comes with constant consultation with professor and the senior lab mates, but we were always encouraged to come up with ideas and solution. So that encouraged me to read more, formulate ideas, and get get into the flavor of research. So how research and problem solving aptitude and attitude can be developed. So that was another key point that IIT Kanpur has inculcated within. Okay. Second, IIT Kanpur offered so we had our department. Obviously, we had departmental subjects, but we had the flexibility to take up subjects from any department along with our department. Okay, so in the meantime, I had I used to take up subjects from chemistry and physics, who are also offering some subjects on crystallography, small molecule crystallography, structural biology. The macromolecular part was already offered by our department, but the other peripheries of the structure and function, or the crystallography, or the physics of crystallography, were being offered by other department. And as that flexibility was there, I could easily choose and pursue those subjects. And obviously, it was not restricted to structural biology or structural chemistry, but it was beyond that. I had a flavor of many, many interdisciplinary subjects. So all of these things, first, to formulate ideas, to solve problems, at least with some help, but we should come up with some ideas at first, and this was highly encouraged. And secondly, we had the flavor of multidisciplinary, interdisciplinary subjects that gave me the confidence and the acne to start or to think beyond my, like it gave me enormous courage that I can, okay, I can deal with any problem. So it may not be restricted, the solution may not be restricted to my field of study, but I'm open to explore. So that was the foundation thing that was inculcated within me from IIT Kanpur. And thanks to them,
Wonderful.
thanks to my professors. Yeah.
Absolutely. And I think here I would like to say to all the audience, whosoever listening to this conversation, having developed this independence, as Dr. Day mentioned, that you are going to be, if you are planning to be a faculty, it is best to have this training early on, where you come up with a hypothesis, the methodology you can develop. And as Dr. Day mentioned, you should use all the help which is there. Either it is your supervisor, your peers in the lab, or whosoever you can ask whenever you face a hurdle or you are troubleshooting. Because if everything is working fine, then probably you are
Yes.
those exceptional ones. But for most of the researchers, it always have a bumpy road ahead. So now,
Yeah.
talking of bumpy roads, let's talk about the journey
Yes.
you had during your PhD in South Korea.
Okay, so that is the most interesting part, and yeah. So definitely, as I said, I started working in a structural biology lab. Obviously, the first project that I was working on was obviously to find and to solve the structure. So first to crystallize and solve the structure of very important proteins. Those were the stem cell transcription factor. And interestingly, those are known as the master regulator of pluripotent stem cells. I can tell you the name. It was one was SOX2 and another was OCT4. These were at that time known as the master regulator of stem cell. What it means is that that when these two transcription factor are activated, it can start a cascade of things within the pluripotent stem cell that helps to maintain the pluripotency. So that is the very basic layman's way of understanding these two transcription master regulator. Okay. Okay. So obviously my professor gave me, okay, you work on these two proteins. These are very important proteins. So once the structure is done, we will have a nice publication and we'll have a many stories unfolding around this. That okay, very good. So I started working. I started purifying. First, the first hurdle, I started purifying, and the purification was very nasty, didn't get purified. And I should tell to your audience that this purification, I think some of you might know, for crystallographers, uh, one of the things we are really concerned is the purity of the protein sometimes, because more purer a protein that we want to crystallize, the chances of getting the crystal it gets enhanced. There are multiple reasons why it is so, but there are also contradictions because people have solved the structure of ribosome, which is not so homogeneous. It is a heterogeneous molecule, but still it gets crystallized. That's a different story. I don't want to go into details of these technicalities. But again, so purity was one of the key thing that we should working for as a crystallographer. So I started purifying. And that purification was really nasty. It was not getting purified. Even if it is getting purified, the most of the purified protein is getting precipitated, not in the correct folded form, in the nice soluble form, because if you don't have a soluble form, you cannot set up crystals. So many things. So I started troubleshooting. I was very nervous. I started troubleshooting, sometimes increasing the salt, sometimes changing the ionic concentration, and so on and so forth, the pH and other things. But nothing worked. At the end of 6 months, after struggling a lot with these proteins, I found that I got a little bit of purified proteins, but that was not good enough to start up setting up crystals. I was a little bit upset and very nervous because that is a new lab. I am not sure how my professors will react. Probably it's my hand, it's not working. I tried out everything. I consulted my lab peers, lab seniors, but nothing helped. I read many papers, nothing helped. And obviously, there is a saying that the proteins, the molecules, which are very important in nature, they are the hardest to crystallize or purify. So there is a myth. I should not say myth, but there is a saying like this. So yeah, it all happened like that. So after everything, I was very upset, and I came to my professor and said, Professor, I am very sorry. I think things are not working for me, and I think I should quit. Quit. I quit even PhD. So I think this is not my cup of tea. I was so frustrated. So nothing is working. So he said, what? No. Yeah, it's a part of PhD. Okay, have a seat, have a cup of coffee. You don't have to be so stressed. It's fine. It happens like that. So don't worry. Uh, why don't you think a little bit more? What can be done around this, or let's try to fix it. If you come up with some idea, just take your time. Don't worry. It's fine. It's only 6 months. It's nothing. You are not losing anything out. So please be calm down, and we can still work and like this. So don't worry, you don't have to quit. It is fine. It's a part of PhD. It also happens. So he gave me all those positive words, and then uh, I again came back to my lab. I started thinking what I can do. It's not working in this way. So then again, I started, okay, if the crystallization does not work, if the protein purification does not work, still lot is unknown about this two protein with context to stem cell, the pluripotency, and many other things. So why shouldn't I start something outside structural biology as a whole? So that was another bold thought, rather I said a wild thought, because I am in a structural biology lab, and now I am thinking I should work on something of stem cell biology. First, I have practically no experience on stem cell biology. Leave alone stem cell biology, I have no experience on working on cells, tissue culture, anything of that sort. And still, I am dreaming that, okay, I will work on stem cell biology, work on some cell biology, and try to solve some problem. So but I was kind of emotional, kind of passionate. I say, okay, whatever it is, I will go to my professor and tell my idea, that I have some very brief idea to work on some other way, circumventing the need of crystallization and purification and crystallization. We can have other routes to solve this problem, to understand these two proteins. So I went to him and said, "That's professor. So these are the things. For the time being, I'm just holding this crystallization part. I put it this in hold. I want to work in this way." Say, okay, great, work, no problem. I have no problem. If you are uh, trying to or ready to take the challenges, I have no problem. But I should tell you, I also have no idea about cell biology or stem cell biology. But what I can do is I can fix you up with another faculty who is also a very famous stem cell biologist in the same school of medicine. She should be able to help you or give you some idea. So I said, okay, professor, that is a great thing. So could you fix me up? So he said, okay. So he called her up, and she eventually came to the office, and we had a long discussion about the things. So that is very important. So it was not only me who was not afraid of taking new challenges, new ideas, but my professor was so supportive. And obviously, the other professor was a very famous stem cell biologist. She was so kind, so helpful. She gave me the next level of courage and said that, don't worry, we will help you. I am also a chemist. Our physical chemistry was my background, and I shifted to stem cell biology. So if I can shift, and I have seen people shifting, so don't worry, it will happen. So I started up setting up cell biology lab for the first time in my lab, because before that it was a structural biology lab working on proteins and bacteria, and to some extent insect cell purification system. But tissue culture and cell biology was never been the main focus of our lab. I started setting up all the facilities. So yeah, it eventually took another 3 4 months to set up everything, obviously with the help of the other lab. But next thing, it started working, and that is the game changer. So finally, I could solve the hypothesis. I could give the strong reason behind that hypothesis I was working at that time, and eventually it made us publish in PNAS. My first PhD paper was in PNAS, and that was also a great big news because we solved a very crucial part about this SOX2 protein. At that time it was not known. Yeah, that's how that shift happened, and I was very thankful and happy that my professor allowed me to do that, and he
Mhm.
always was very encouraging in the entire
Absolutely.
journey.
I think one of the things during these conversations which come up often that when a young enthusiastic researcher join PhD with all full passion, they just want
Yes,
to do all the things which sometimes are tough, sometimes very close to impossible. But because they are naive to the situation, so they
yes,
think that it can be done. But
yeah,
having that support of your supervisor, and as in your case, even if the mentor himself or herself is not the expert of the area, but setting that up, giving that support, that they have someone else in their network who can support you and give you the wings wherever you want to fly, is very important part. Like whenever you are choosing your PhD labs, having this support from the mentor, and if you can have this conversation with them before, like what happens if you want to do something tangentially with the lab research focused, because that is what happened in Dr. Debojyoti's case. And also it is not very rare because this happens all the time, especially now when we started talking of interdisciplinary research. So a person from quantum physics is working with a biologist using chemistry and whatnot. Like for now biology cases, people from different stakeholders, even civil societies and whatnot, all everyone is coming together to make the world the better place, and researchers play a very crucial role in that. So Dr. Day, having done your PhD, like just
yes.
before changing your name from Debojyoti Day to Dr. Debojyoti Day, what were the
Mhm.
plans before that ceremony? Like what you want to do with that degree?
Okay. Um, Obviously, at that point of time, my path was set. So I will, because another thing that I should mention here. So during my final years of PhD, my professor also involved me taking classes. That is not a very, I think, in most of the universities outside and also in India, that is probably a routine thing. But by the end of my PhD years, I took like I started taking most of the classes. Like I had the courses were registered in my name at that time. It was not like my I am taking my professor's class. It was not happening in that way. My professor made the School of Medicine register the courses in my name, and that is another very interesting thing that shaped my the journey, the future of journey ahead. So during my those teaching time, I figured out I love teaching. I love telling stories. And again, it started from my school days, how my teachers used to tell stories, how it could inspire people. So once I'm in the classroom, I'm talking about my research or research of my peers, I was the happiest person. Okay. So I am stress free. I am free of all kinds of thoughts around me, around this world. I'm a very happy person inside the classroom. So that is how I realized. So this is the thing I am going to. I'll teach. So that is the another reason. The first when I was looking for position in India, I didn't look it in a research lab. Rather, I was looking in an institute, educational academic institution to come back because the teaching is it should be I thought of at that time would be a primary parcel of my uh, the next journey along with my research like 50-50 or 40-60 like something like that. So That is the thing I decided at that time, that I'll be teaching. Also, I'll be doing research, and I'll be inspiring new minds. That is the thing. So at least we should work together, make new uh, new battalion who would work with me, teach them, and make them more able, and we can solve problems together. So that was the idea. Yes.
It's like keeping the idea of giving back to society. As you were
Yeah, correct.
inspired by your school
I
teachers,
would
now it
I
is
would
your turn.
I would rather say it was more than that. It was like I was the happiest person when I touch.
Mm-hmm.
So in the back of mind, that was the thing happening always while taking that decision. So whenever I am in the classroom, I'm talking about my research. I'm very happy. So why I should think some other way? So this is the best way. This is the only thing I can do. So let's do it. So that is how it's
Absolutely.
happening. Yeah, yeah.
So having that faculty tenure track position in South
Mhm,
Korea, the the infrastructure you had in South Korea, and when
mhm,
you moved back to India,
mhm,
because when the first time when you moved from India to South Korea for your PhD, you were this, passionate researcher who want to achieve so many milestones, going to do new discoveries, because as that was the group you joined. So you had those aspirations in there. You had experienced that for extended period, and then now moving back to India, any challenges you faced when you came back, either it is about infrastructure or it about the different societies, you know what I'm talking.
yeah. So, for that, to answer that question, I would take you a little bit back while I was doing my tenure track and all those things. So, as I said, my professor was very supportive to try out all interdisciplinary stuff. So, eventually, during my PhD, post PhD, and my tenure track, also in my as a postdoc, I worked on various interdisciplinary areas. Obviously, I was aiming for the same research problem, but solving my research problem was not restricted to any particular discipline. So, as I said, I started from cell biology, stem cell biology, worked on drug delivery, worked on uh, the biophysical part like atomic force microscopy, electron microscopy, worked on animal models, because obviously to testing hypothesis you need to do those things. Animal models was also not restricted to mice and hamsters, but also big animals like monkeys, rhesus monkeys and all. So, and obviously, I was actively collaborating at that point of time with many different groups who work on different disciplines within that country, South Korea, outside that country, in Japan, Germany, and many other places. I used to visit their lab, learn new things, come with those technologies, and actively collaborate. That was a wonderful learning experience because I was exposed to various different fields of study. Okay, so definitely I always had that idea. Probably I will not have that much flexibility when I am coming back to India, because obviously you are primed to different kind of stories that okay, you will get restricted. There are not things working in that way. I said it doesn't matter. I will anyway come back. Let's see. I'll figure out. Okay. But then when I come back, so I joined this institute at 2019, because 2018 I came back at the end of the year, and 2019 January I joined this position. And as you know, thereafter the COVID breakout. Okay, I had a few projects in my hand. It was funded by the ACRB at that time, and ICMR. That there are two projects, but definitely those projects were mostly into the wet lab experiments and all those things. But as the COVID started, I couldn't work on any of them. Okay, so again, it gave me another challenge to change my Okay, before that, I should answer your question. Was the infrastructurally or funding wise or mentality wise, did I face any such issues? I should say yes and no. It happens in both ways. So I cannot say everything was bad or everything was good. It was all the part of the life. It's the flip of the coins. So something worked, something didn't work. If something didn't work, I found another way of work. So that's how I should put it. Okay. Funding was not a problem at that time. Still, it is not a problem because as long as you have ideas, you have something to fund your research. That's another thing. So this saying this too, then the third thing is yes, then the COVID came. So what can be done next? I started telling my PhD students, see, there is COVID now. At least from 1 year down the line, you cannot work in the lab. You cannot do whatever has been planned. So let's try out one thing because I don't want to delay your PhD or delay my research. So let's try exploring the computational biology, the machine learning, the deep learning, all those things. Again, definitely I had little bit background as because I had a BTech degree, a little bit coding and all, but that was insufficient to make a decision that we will be doing computational biology all of a sudden. And I should say, tell you, this was not restricted to simple docking and all. We were writing codes to analyze certain parts of genome, so understanding the genome or the transcriptome, or rather omics better than what was known at that time. So it was not like docking and all those things that people do generally in the name of computational biology. So I was actively looking for those kind of application where we should get some idea to solve problems or design problems right from different levels of omics. And for that, we started looking for understanding patterns and pattern recognition, understanding pattern, to work with that pattern, to understand, to connect them with context of biology. Again, I started reading, I started training myself with codes and all those things that are needed. And also I started training my students. Again, those same ideas that because my professors were not afraid to take up new challenges, to learn new things. So why should I be restricted to whatever I know? I should also open that entire world of challenges to my students and let them figure out, let them learn. I also learn in the process. Again, I shifted from those stem cell biology, regenerative medicine I was working on, to obviously the main focus on stem cell biology and regenerative medicine. And also later came the metabolic disorder, because that was a problem of Southeast Asian countries at that, means still now it's a problem, the metabolic disorder. So those things added up, and we started working on computational ways of dealing with them, to formulate new ideas, to solve problems, and how to align them to my idea of understanding the regenerative medicine and stem cell biology, and how this can be solved by other disciplines like computations and coding and all. So that's
Absolutely.
how it started. And my
Mhm.
first PhD student, that he has already graduated, his work was completely computational biology.
Nice.
He but he started and joined the lab to do some wet lab experiment. But at the time he was graduating, he said, "Sir, thanks to you, I don't regret. It was a fantastic journey, and it was wonderful because I learned a lot of things. And the most important part, I could finish in time, because most of the COVID batch, they couldn't finish in time because of that lag. They got stuck. Their fellowship were over,
Mm.
and they had a very hard time completing
That
this.
is what we always say, that you showed this once again, that Scientists, the researchers can convert any challenge into an opportunity and then open the Pandora box where people will do research for next decades. With this, we'll say thank you so much, Dr. Day, for taking time out. And for all the audience, we'll say that if you want to connect to Dr. Debojyoti, then you can just write to him. If you want STEM for Society to put you in touch with our researcher celebrities, we are there to support you. What all we want is, if research makes you happy, we are there to support you in all the way we can. Thank you so much, Dr. Debojyoti, once again.
Thank you. Thank you. My pleasure. My pleasure.
To all the audience, we'll say stay tuned for another episode of Researchers Celebrity.